Bismillahirrahmanirrahim. Assalamu alaikum to everyone. This is Muhammad Zawam Ali, and today I'm going to present a topic which is DNA sequencing techniques. What is a DNA sequencing? Sequencing means finding the nucleotide order on a DNA piece, which can later be used to determine which determine the amino acid order. What is the order of the amino acid? And when these nucleotides are extended, they form different kind of protein structures which are responsible for various kind of function. This study deals with the proteomics. Or and DNA sequencing is very useful as when alteration is DNA or alteration in DNA sequence can lead to an altered or non-functional protein, which had an harmful effect on the animals or plant also. A DNA sequence is very important as if we don't know the DNA sequence, then we won't be able to tell either some kind of disease is due to mutation or something else. Understanding a particular DNA sequence can shed light on genetic conditions and offer hope for the eventual development of treatment. And it is very important for the for treatment for curing any disease, especially the hereditary one. Not only this, DNA technology is also extended to environmental, agriculture, and forensic applications. General principle of DNA sequence is that it synthesizes various kind and various length of DNA subfragment, and we separate them on gel to get the desired band, and on these basis we determine the sequence of our DNA. Therefore, if the template or if the template were 200 base pair in length, there should be 200 different subfragments ranging from 1 base pair to 200 base pair. This means that if we have a 200 base pair, then there should be 200 fragments uh, having different lengths, different sizes like that one base pair and other 200 base pair. DNA sequencing methods include Sanger sequencing method, which is the most old method, which use dideoxynucleotide. Both of the oxygen on two carbon and three carbon is removed, and it is also done with the chain termination method. Its accuracy is very high, around 99.9%, but it is very costly. For 1 million base pair, it costs around $1,000. Then the other method is the Maxim and Gilbert sequencing method. It is same like the Sanger sequencing method, but the only difference is that it uses chemical sequencing or chemical termination method. Some advanced DNA sequencing methods include short DNA sequencing, and the next generation DNA sequencing include Illumina sequencing. Illumina sequencing accuracy is very high, about 98%. It is accurate, um, and not only its accuracy is high, but it is also very cheap, which is 0.05 to 0.15 cents per 1 million base pair. And last but not the least is our pyro sequencing. Okay, the guy is Frederick Sanger, who was an Britain biochemist, discovered DNA sequencing by chain termination method, and he first won the Nobel Prize in 1958 for completing amino acid sequence of insulin. Later on 1980, he won another Nobel Prize, which was for DNA sequencing. For illustrating the Sanger method, we are considering an example having a DNA sequencing of 8 base ACG, ATT, AG. Uh, for Sanger sequencing, we must have a template, a primer, and radioactive DNTP for all bases, for all four bases, which were adenine, thymine, cytosine, guanine, and DNA polymerase, so that our template for our sequence can be extended. Uh, and these all are mixed together. Uh, this mixture is distributed or divided evenly on four tubes, four Eppendorf tubes, each having a different dideoxynucleotide. One may contain DD, TTP, DD, TTP. One may contain DDGTP, DDCTP, DDATP. Okay. Since the largest DNA fragment generated by sequencing technologies are only 200 to 300 base pair in length, so therefore we use the polyacrylamide gel because agarose with this small size is very difficult to carry on, or we can't dissolve these with the help of agarose as it contains a very large pools. Okay, this is an example illustration of the Sanger sequencing. This contain our all master mix primer and our DNA polymerase, etc. But the only difference with all of these four Eppendorf tube is with DDATB, which is a dideoxy uh, nucleotide, holds all oxygen on both 2 prime and 2 carbon and 3 carbon is removed, and TTP, CTP, and GTP. Uh, when dideoxynucleotide is added on the growing DNA strand, and it will terminate the further extension and the reaction is stopped. The Sanger method includes for Sanger method, there must be a large number of multiple copies of single-threaded template DNA. A suitable primer should be there, which pair with the template DNA and start and act as a starting point for the replication. DNA polymerase, which incorporates our new nucleotide at a 3 prime end, a number of normal nucleotides which are ATP, GTP, and adenine, thymine, cytosine, and guanine, and a small portion of dideoxynucleotide, which may be radioactivity level or fluorescent dyed level, and the template DNA. The template DNA pieces are replicated, which incorporate nucleotide. But sometimes we use the random deoxynucleotide for this purpose, which stop the growing DNA. Uh, as a result of dideoxynucleotide, all the DNA length end with a particular label dideoxynucleotide, as it lacks both the oxygen on 2 carbon and 3 carbon, so no further extension is not possible and the DNA polymerase is removed. As we know this, that in gel electrophoresis we have a different size, a different size of DNA fragment. So some are possibly travel to a long or larger distance, um, or move very fast, which have a very small DNA, and other is moved slowly, which have a large DNA sequence or their size, or and those having a large size. Originally, four separate sets of DNA, primer, and a single different dideoxynucleotide were produced, and these were run on the gel. Modern technologies uh, help us to mix the DNA and primer, and help us to fluorescently label dideoxynucleotide in spite of which can be treated easily with the help of laser. Uh, More Additionally, we in the modern technology we also remove the gel slab and it was replaced by the polymer filled capillary tubes. Okay, this is an example. This is a ribose sugar which contain both oxygen on its two prime and three of two carbon and three carbon. It is a deoxy, which means one oxygen is removed on the two carbon, and it is a dideoxy where uh, oxygen on the both two carbon and three carbon is removed. Okay, this is an example of uh, Sanger sequencing. Just consider this as our DNA sequence. As we know, we yet have to sequence our DNA fragment. So, for developing an understanding or to understand how this thing works, we made an random sequence. As we know, the sequence is not that much small. It is generally 250 to 200 to 250 base pair long. Okay, and this is an engineered sequence so that our primer can attach. This is our primer. Okay, this is our dideoxynucleotide. As we can see, this is for ATP, and we also know that A is A always bond with T, or it is a complementary to T. So anywhere in the sequence, when we get AT, it is replaced by the A. Same for this, again same for this. It will be our first sequence. T is present on the edge, so our reaction is started by the but terminated on the same end as we added the DT-ATP and it will detect the T and terminated the whole reaction and the first sequence will be our A. Then we can see T is complementary to A is complementary to T, C for G, G for C, and T for A. As you can see, this is exactly the same sequence which I told you earlier, that T is complementary to A for T, C for G, G for C. Then A came again. It is because T is there and T is complementary to A. When they found uh, complementary to A, then the dideoxy nucleic acid terminated the whole reaction. And the same exactly uh, for this. This is for DDCTP, as you can say, when the G detected when the C detected G, and it's complementary. G is complementary to C, so it causes the termination of the reaction. And this is again for GAC. As you know, C is complementary to G, so C two fragment is produced as C is there and C is there. This is for TTP, exactly the same as I told you earlier. Okay. Once the sequence is complete, and different size and different length of fragment is produced. As you can see here, it has an size is 1. It contains 4 nucleotides. It contains 3, and it contains 2 with different sizes and variation. Then we transfer our DNA sequences to polyacrylamide gel electrophoresis for the visualization. As we know, these are different bells, like this is for adenine, this is for thymine, cytosine, and guanine, and we put our sequences like which was those which was for DT, ATP, uh, in on this well, and those for TTP in this well, and this well. As we know, in gel electrophoresis, larger fragments move slowly as compared to smallest fragment. So a larger fragment will be there, and a small fragment will be there. After this, we do autoradiography of our gel, and uh, this is different kind of sequence which we were obtained. Okay, this is a basic. This is a very core concept and very important term here. Is that as we know, the largest fragment move from this direction to this direction. Largest fragment were present there, and small fragment were present there. So, uh, basically, in this diagram depict the DNA sequence. So we can find that we can start our DNA sequencing from 5 prime to 3 prime, from bottom to top. As you can see, A is there, T, A is lightweight, then T, then G, then C, then A is lightweight, then C is lightweight, and the vice versa. This is our obtained sequence after the reaction. This is another example of real sequencing gel. Um, Nowadays, with the advancement of the technology, we have a dye sequencing technology where we use a dye in spite of using uh, radioactivity label isotopes. Okay. Uh, DNA fragments were originally detected by radioactivity labeling. We primarily use radioactive labeling, but nowadays frozen dye method is commonly used. It also contains four different labels, so that we can label our with these labels we label our four nucleotide chains with their different dye, so that it is easy to measure and observe. Uh, these dyes fluorescence are a unique wavelength, and it is only observed when the specific wavelength is present. One of the major advantage of dye sequencing is that it is not very costly. It is very easy, and cheaper. Okay, it is a sample dyeing. Sorry, it is a dye sequencing output. As you can see, A is labeled with green color, T with red, G with yellow, and C with and G with yellow and C with blue. When okay, when there is A is detected, it will emit the green color. T is detected, it will emit red color, and so on. It is another example. It is a DDK reaction. Okay, and here we get our sequence. As we can see, A is labeled purple, G is pink, and so on. So what is going there is a laser beam is emitted there, and due to laser light, an electric current or impulse is produced. So this impulse is detected on the detector, which is attached with our computer, so we can find the sequence. It is an other diagram. As you can see, this is exactly flat sequence which is coming here. Then we can see the slight mountainous region, and the topography is changing, some with down, some with up, like this. Uh, Basically, this spontaneous region indicate the intensity of our nucleotide. As you can see, this is a very much up as compared to this flat region. It means that the frequency of C is very much higher as compared to T on the initial phase of DNA sequencing. The modern technology. mm, uh, helps the scientist and the other communities or with the use of bioinformatics um, in solving the in solving the biology problem had a significant role. Nowadays, we don't even have to read the sequence from the gel. There is no need. There is no need to sequence the. There is no need to sequence from the whole gel. We just the computer did this for us. This is a plot of the color in the next line, which is detected on one lane of a gel or one sample scanned from smallest fragment to largest. The computer interprets those colors by printing nucleotide sequence across the top of the plant. And the next method is Maxim and Gilbert chemical sequencing. This chemical sequencing is based on the ability of base-specific chemical reagents. In this method, it is basically same method as we have Sanger method, but the only differences arise with chemical. The only differences arise on the base specific chemical reagent, where a specific base or a specific nucleotide is removed when they are treated with the chemical. Four sets of deoxy oligonucleotides are generated when subjected to purified 3' or 5' and labeled deoxy oligonucleotide were base-specific chemical reagent. They randomly cleave at one or two specific nucleotide. The demonstration is given below. Uh, for base-specific chemical reagent, we need a hydrazine dimethyl sulfate for G. For C, formic acid is needed to remove the G and A, and then piperidine is added to cut the sequence, to cut the region where the DA, where piperidine is basically used to cut that specific region where our where sequence where our base is or nucleotide is removed. In the first reaction, what we do, we use hydrazine, DMS, or formic acid uh, for removing bases. Then on the second half, we use piperidine strand for the quantitative cleavage for cleaving the DNA fragment, so that sizes different sizes uh, of DNA fragment is obtained, should obtained. Okay, this is an illustration or diagram of Maxim-Gilbert method. First of all, we have a sample DNA, which is a DNA which is double stranded. Then what we do, we make it a single stranded DNA and make it a homogeneous. Uh, After this, we add P32 radioactive isotopes as a 5' phosphate and cleave at specific site. Um Actually, this is a very important concept, or which is the heart of the Max-Weber method. Now, look at this. We have first of all we have a G reaction. It is a G reaction. It means that wherever in this sequence, when the G is found, this DNA, this G reaction, this D nucleotide is removed by DMS. DMS removed the G. Wherever in the sequence of DNA is found. Okay. Uh, as you can see, we have an ATT there. ATT is obtained. Right. No, God. ATTG. When the G is removed, we have an ATT left. Then we have ATTGSETTA, ATTGGSETTA. Then we have a G there, and it is removed. And other thing is A reaction. Uh, in this DNA sequence, wherever the A is present, A nucleotide will be removed, and sometimes uh, G is also removed there. After this, we have a T reaction. Wherever the T is present, T is removed with uh, frequently or occasionally. C is also cleaved. After this, we have a C reaction. Wherever in the sequence C is found, C is removed. C will be removed. Okay. When all the nucleotide or when the nucleotides by with the help of specific chemicals are removed, we find such kind of sequence. It is a sequence which we obtained. Then we put it on, or we do electrophoresis of this DNA sequence and observed on radioautography. As you can see, we have an fragment length or basis. Then we have read sequence. Okay, this is basically a whole nucleotide. This is basically a DNA fragment which was obtained, and its size is 12. It is a 12 base pair long. It is present on all the four nucleotides well because the sequence length is 12 base pair. After this, we have on 11 we have two sequence where this is a very dark sequence and this is comparatively light sequence. On 11, we it's mean we on 11 sequence number 11 we have what we have a C cytosine. We can also see this on exactly 11 we have another sequence which is T reaction or which could be T or C. What does this term indicate? Look, we don't have any one sequence, or it is not possible that we have a C and T on the same place. So, how can we find this is a C? Because on the 11 on the fragment size we have on 11 size we have a C, but on 11 we also have a T and C. So this means we should have a C there because it possesses an ability to cleave the C and T. And so there we have a C. After this on 10 base again we have uh, C sequence and T and C sequence. So this is exactly the same as it is not possible we could have two nucleotides or two bases on the same size on the same place. So again, its mean it and it is although it is its major portion is to cleave the T, but it cleaves the C again there. On 10, it cleaves the G, and we can see the band here on 10 base pair again. So it means it cleaves the G, and this thing goes on. The chemical mechanism of the reaction is follow. The shaded base at the C prime end of the fragment to the right of the gel indicate bases that have been chemically modified. Those bases which are chemically modified were existing on the C prime end. Then fragment of these bases can be displayed by piperidine after each cleavage. Cleavage is generated by piperidine and it generated different. fragment. As you can see, after a limited reaction with DMS, which is specific for Gs, it means uh, Gs were removed with the help of DMS, and then these residues were cut by piperidine. Uh, then uh, Following method is used for cutting the G's and A's. Hydrazine is in the absence of NaCl. Basically, hydrazine in the absence of NaCl cleaves TC lane, and hydrazine in the presence of NaCl cleave only C's. Thus, a band is observed in the C lane. Another method is shotgun sequencing, which is in kind of a modern method where we in shotgun sequencing the genome is randomly broken into short fragments. We break the genome into small fragments, which is generally 1 to 2 kb long and suitable for sequencing. Then these fragments were ligated into suitable vectors. The vectors are required in this method, and when they are inserted into vector, then we partially sequence them. Around 400 to 500 bases of sequence were generated in the single sequence run on the first phase. This first phase, as we know, uh, due to shotgun sequences, as it is random and has an overlapping region, so it is not specific like this. Uh, we found such kind of sequence. So we use computer for these overlapping sequencing for easily assembly, and so that we can get the exact sequence in a single row. Uh, for gene gun or for shotgun sequencing, sorry, for shotgun sequencing, no genetic map or prior information is needed about the organism whose genome is TB sequence. We can easily uh, sequence the DNA. The limitation which was concerned with shotgun sequencing earlier was that it is very difficult to handle this massive data, but with the advancement in technology, fast computers were used now for this problem. Uh, Now, for cloning vector types, we have plasmids, which are generally up to 5 kb, which can store DNA having size of 5 kb. Page lambda is up to 50, bag 300, and yak 2000 kb. Another sequencing method is which is relatively advanced method is next generation sequencing is Illumina sequencing. As we know, DNA is in double stranded condition, so we first have to make it a single strand DNA, which we bound it to the flow cell channel. Then this flow cell contains large number of primers, all of its surface. As you can see, this is then a flow cell and contain this minute or tiny or small size primer. Then the next step is prepare genomic DNA sample. We can see this is our double-stranded DNA. We have an adapter there. We must ligate them on DNA on the both sides. Fragment DNA of insert into smaller fragments that are able to be sequenced. We make uh, of our DNA, of our large size DNA, we cut it into the small fragments so that we can easily sequence it with the help of sonication, nebulization, and enzyme digestion. After this, we ligate the adapter and denature the double stranded at 95 degrees centigrade. Once uh, it is attached with the primer, it form a large sequence like this. We have beads there. Once the sequence is completed, then it is band like this. Unlabeled nucleotide and polymerase enzymes are added and, and to initiate the solid phase bridge amplification. Bridge amplification means that when they are finding the similar color on both the tails and the head, then the sequence which is already completed is bent towards the same sequence. Like this is purple, it will be bent always towards purple, and the red is always bent towards red. Uh, After forming bridge, the later on bridge was become double stranded because primer, nucleotide, polymerase enzyme, and buffers were added there for completing its sequence. Uh, After this, the next step is denaturation of double-stranded molecules. The original strand uh, is washed away, while the and that has been synthesized to the oligo attached to the floss. Then we repeat the step 5 and 7 again and again until we had a cluster of fragments. Cycle of new strand synthesis and denaturation to make multiple copy of the sequence. Now, then our fragment become double-stranded and we denature it. Okay. Once the reaction is once all the sequence are made, then the next step is determining the first base or insertion of first base. Uh, Sequence reagent were primers, our polymerase, fluorescently labeled nucleotide, and buffer. When these all things were completed, then our first base is incorporated. Image of the first base, we remove unincorporated base. Uh, when our first base is inserted, it detects or it release some kind of signal. So we deblock it and remove the fluorescent signal, and the next reaction is initiated. Then the second base is added in the same sense. Primer, polymerase, fluorescent nucleotide, and buffer add sequencing reagent, and the second base is incorporated. There's the same for image for second base. Okay, after the sequence, read over multiple chemistry cycles. Then the identity of each base of a cluster is read off from sequential image. We can, with the help of these image, we can find what is our sequence of our bases. Is As we can see, each base is labeled differently, and it gives us the pop information. As we can see, uh, and the data is very huge or very large, so we don't have exact DNA sequencing. Our the sequence which we obtain is generally overlapping like this. So we use algorithms, or we need a fast computer for helping us there. So we align data with the help of computers. Okay. The next method is our pyrosequencing. It is also a new method where we use pyrosequencing is based on the generation of a light signal. A light signal is released uh, when the DNA, when the DNTPs or nucleotide is added in the DNA. Or basically on the three carbon we have a three phosphate. So every time when there is a nucleotide, when there is addition of nucleotide, two phosphate group is released, which is a pyrosequencing. Pyrosequencing is pyrosequencing or pyrophosphate. Pyrophosphate is used to generate ATP. Okay. Uh, with the help of adenosine phosphosulfate, pyrophosphate produce ATP. Then this ATP and luciferase enzyme generate light by converting luciferin to oxaloferrin. Uh, in this method, each nucleotide is added in turn, which means that at a time we can add one nucleotide uh, at a time. Only one of the four will generate a light signal. The remaining nucleotides are removed enzymatically with the help of enzymes, and it is recorded on the pyrogram. The a flow diagram, or basically the chemistry of pyrosequencing, is let us consider this is our template DNA. We have to engineer it in some way so that our primer can attach and we can initiate our reaction. So, what does this indicate? DATP alpha S is exactly the same as we have at DATP. The only difference is that uh, luciferin generally use ATP, but DNA polymerase uh, have an potential to use uh, a DATP either in the to use ATP either in the form of DATP or DATP alpha, while the luciferin can use only in the form of ATP. Okay, as we know, G is always pair with C, but what we have there, we added DATP, but as you know, G can't pair with A, so there is a no emission or no light is produced. The light is produced only when complementary sequence bind with each other, like this. As you can see, G is pair with C, and it reduces to uh, pyrophosphate or to phosphatase, and the light the light is released there. Okay. Uh, this means this means that G can't pair with A, so outcome is negative. But G can pair with C, so outcome will be positive. It's and it can't pair with again A. G can't pair with A. It's negative. Okay. Uh, the same thing continues. This is an output of what we have there. As G can pair with C, so uh, G can't pair with A, so we have a negative. G pair with C, we have positive. A can't pair with G, so we have a negative. A pair with T, so we have a positive, and the same thing continues. Uh, at the end, we have an application of our sequencing. We use it in forensic for identifying the individual on the basis of different genetic sequence. In the medical field, we use it in muscle dystrophy for diagnosing. We use it for mutagenic disorder diagnosis, and in agriculture for mapping and sequencing of genome. Uh, Thank you so much for your time and patience.